All right. All yours, Chair.
Okay.
All yours. He's going to introduce.
All right. For agenda item number 7, we're very lucky to get a presentation today about blue-green mitigation strategies and technology presented by Dr. Barry Rosen with the Florida Gulf Coast University. So the purpose of this presentation is to share some of the latest research findings regarding cyanobacteria mitigation techniques. And the connection to the CCMP is that this relates to Water Quality Improvement Action 5, that's to support harmful algal bloom or HAB, including microalgae research and monitoring and measures to proactively reduce the drivers to the reduction of anthropogenic nutrient pollution and other contributors.
All right.
Thank you.
Well, thank you so much for the invitation and letting me come and talk about one of my favorite things. And let's, first of all, I'm with Florida Gulf Coast University. I'm a world-class scholar and professor. So 7 years ago, they hired me and I left my 20+ year career as a federal employee. I worked for NRCS initially on water quality related things in watersheds. I worked for U.S. Fish and Wildlife running Everglades Coalition, Everglades Restoration out of Vero Beach. And then USGS said, we want you to run the Florida Integrated Science Center, which was all of USGS in Florida, 400 people, $50 million, way too much meeting going. I call it administrivia, but I was on a lot of the boards that had to do with running Everglades Restoration. I really like my current job so much better. Anyways, that's the caveat. Oh, point at the thing there. All right, so first of all, why do we call them blue-green algae and why are they so successful? I bet you this is a pointer here, no? Is there a pointer built into this?
Yes.
Okay, so blue-green algae, like all algae, algae itself is not a taxonomic term. So when I say algae, it includes things that are bacteria, the cyanobacteria, and it includes green algae, includes sargassum, red drift algae, Kernia brevis for red tide. All those are algae, and they're only united because they all have chlorophyll A. Now, some of the algae have other pigments that make them look bluish green. And here is the cyanobacteria. Here they've got, here's the chlorophyll A right over there, absorbing at a couple of different wavelengths. But they have phycocyanin over here, which is that bluish color. You see the word cyan in there, and phycoerythrin, like erythrocytes, the red blood cells. Those two other pigments allow them to absorb light more than just chlorophyll A. They can get different wavelengths because as water, you know, as the sun hits the water, certain wavelengths get filtered out quicker because they're bigger, and the shorter wavelengths can penetrate deeper. So they're very effective at capturing sunlight. Whoops. Hey, I didn't do that. I wanted one. Go back. Can you have it go back? One more. So this is an interesting thing, is that these pigments don't make them all blue-green. There are some that look black, just a mixture of pigments. The organism on the left is a cyanobacteria, but it looks black. And here's one in the middle that looks very bluish green, which is right. And again, they have a special molecule inside that captures that sunlight. It has to pass that captured sunlight onto chlorophyll A. But just a little background on why we call them cyanobacteria and why they're well adapted. They also want to maximize the capture of sunlight. And many of the ones that cause blooms have a specialized structure inside the cell. See that structure outlined in yellow? That's called a gas vesicle. Can't be a vacuole because vacuoles are membrane bound. Go back to biology whenever you had it. Membrane bound organelles, that's eukaryotes. This is a bacteria. So it does not have a membrane bound, but it's got this vesicle in there that fills with air, brings them up in the water column. Oh, just the right place to capture the right amount of sunlight. You don't want to get burned out by having too much sun, and they don't want to have too little. But as the photosynthesis happens during the day, they make sugar. They make glycogen, which actually collapses that gas vesicle and makes them go back down. So they basically go up and down in the water column. So think about this when we talk about treatments. If you're at the top of the water column and you're spreading something right on top, you might get them. But part of the population is way down below in the water column. You're not going to get them. The other weird thing, and don't forget these are simple bacteria, is when they get down to the lower part of the water column, a lot of times nutrients are released from the sediments. So they are going up and down, not just to maximize photosynthesis, they're also picking up a load of nutrients when they're at the bottom of the water column. Not all cyanobacteria can do this, but the planktonic ones can. Go forward. Oh, this one. So why do things bloom? And, we don't know the answer. It's been studied for 60 years. But think about this scenario is that you have all these organisms, I depicted them here in different shapes and colors, and there's something that's limiting the key organism that wants to bloom but can't. So what happens is that key element is brought back in and it allows them or that particular organism to bloom. And what is interesting, it's more complicated than rocket science because you have daily changes, weekly, monthly, seasonal. You have every single nutrient with its optimum, every temperature optimum, and each organism integrates all this, again, simple organisms that allows it to bloom and become the dominant and a big problem. Oh, what happened? So first of all, the main organism that people care about and hear about is microcystis. And it's italics there because that's what we do for genera of organisms. But it doesn't mean it's making toxin. You hear of the toxin microcystin. So when I say microcystis, you don't know if that particular organism is a toxin producer. Even if I knew exactly what it is, which is what I do, I don't know if it has the genes to make the toxins. There are strains that do and strains that don't. So that flotation that I was talking about, the gas vesicles, it's kind of interesting that you can actually see them. See the little shiny grooves? Come on, show up. I'll go over here and point to it. See these things here? Those are the gas vesicles. I'm seeing them under the microscope, but the idea that these populations go up and down in the water column is really important to management techniques. So the problem is that the blooms just don't stay put. They don't just come up and stay in one location. These are small organisms, highly pushed around by wind and currents. So if you have 100,000 cells and you've got a toxin level, the buoyancy means that upper layer gets really thick with organisms. And then if the wind blows, they will accumulate on one shore. And the next time you go out to sample, even a few hours later, they could be somewhere completely different. They move all around very easily. They've got big sails on them. They also grow so fast. They're thermophiles. They love warm water. Florida's a good place for them. So in the morning, you could have one cell, and if you have the right temperature and everything else, all those other perfect nutrients, you can have 8 at the end of the day. So when you go to control, keep in mind that you're not in a swimming pool. You can't kill them all. And if you're only killing part of the population, the rest is growing and taking advantage of the space that was made and the nutrients and everything else that became available when you killed part of the population, but not the whole thing. All right, toxins. Very briefly, the main ones we talk about are the liver toxins on top, the hepatotoxins like microcystin, the neurotoxins, which affect the nerves. Then you have skin toxins. BMAA is a potential toxin. We don't really know enough about it yet. And a whole bunch of other compounds we just don't know that much. In freshwater systems, when you have a fish kill, it's because of low dissolved oxygen and not the toxins. Why? These things need oxygen to burn that sugar that they've accumulated during the day. It's called respiration. They're doing it during the day, but you've got oxygen they're making during the day. Oxygen, by the way, is an absolute waste product. Algae don't want it. They spew it out, and I think humans and animals in general want that oxygen. But the low dissolved oxygen happens when a room, let's say this size, has got so much algae in it, the fish can't get away from it, causes a dissolved oxygen sag, and they die, especially at night. All right, enough of the toxins. You know that there are guidelines for toxins, right? You've got drinking water guidelines that came out of the Lake Erie event in Toledo. So you've got guidelines like this: 1.6 milligrams per liter, 3.3 milligrams per liter for microcystins, but for children younger than school age. So think about this: a tap in your house, and you go, "What? Younger than school age? How do I know?" And then it's a 10-day exposure. It's a weird assumption. And again, these are guidelines, not regulations. No drinking water company in the country has to follow these, but they should. Recreation is a little different. That came out in 2019, a much higher concentration. And why that happened is people were boating and swimming and kids were playing on the edge of the water. And they actually did studies where they were watching how much chlorine is excreted from pools and kids. And they said, okay, let's be higher concentration, but it's instantaneous. So if you go measure toxins, that's when you see Department of Health posting, it's the presence of blue-green algae, whether it's making toxins or not. All right, control methods. First and foremost, can you stop the nutrients coming in that feed these? They are just like plants. They need nitrogen and phosphorus, except for the nitrogen fixers, which I won't talk about. So the best management practices, can you get the nutrients low enough from the watershed to have an impact? And, you know, maybe you can do something, but you have to do that in conjunction with whatever short-term measures you might want to do. The second thing, in a water body, especially one that isn't flowing, because if it's flowing, you're going to dilute anything you add to it. You can do algicides. Copper has been used in many different forms. You have cutrine and Captain XRT. It disrupts photosynthesis and kills the organisms. You have peroxide-based, hydrogen peroxide-based compounds that are out there. Green Clean Pro. Again, you break down the organisms. What happens to all that stuff inside the cells? It feeds bacteria. Are you going to have a low dissolved oxygen event when you use this? Very likely, from killing organisms and all their internal parts degrading. And there's other products you can shade the water itself. I took a picture of this one place in Myrtle Beach. The whole lake was blue, aqua blue from aqua shade. Again, what happens to the dead organisms? It's not a swimming pool. You can't really clean them out of there. If you have a water body, again, if it's stable, cyanobacteria don't want to be moved around. They want to be able to move up and down and regulate their own buoyancy. So you can have circulation devices, lots of them for destratifying them. Probably less so in Florida, but you do see a lot of ponds and home communities that have these circulators that circulate the water. It's a good thing. They can't regulate their buoyancy. They're in the shade more than they're in the light. They are probably the most effective way of doing it. Bubble curtains. It's like that. Just, I don't get it. These things are much smaller than a bubble. How can they not pass through a bubble curtain? I know some people swear by trying it. You have to try something. And maybe at the mouth of a canal where the water's coming in, let's say from the river system, which has a load of cyanobacteria, maybe. But they're small. They're going to get through that bubble curtain. And what happens when the power goes off? So you can also do this. There's techniques for sonication which you're breaking the cells with ultrasound. Again, what happens to the dead organisms? And if you are not in a very confined space, like this golf course pond or this community pond, you're really not going to do much good. Also, in red, you see on the left, you didn't eliminate the nutrients when you break up the cells. All you're doing is allowing a recycling path to go on. Nanobubbles were promoted for, probably 5-10 years by a company, several companies, but a couple in particular. I went in with a company to look at the Pahokee Marina. We installed this nanobubble generator. These are little tiny microscopic, submicroscopic bubbles that they claimed that they could fill with ozone. Sounds good. Ozone's a good good oxidizer. It really didn't work at all. Even in these little back areas, there's too many ups and downs, electrical. These are very sensitive machines. And now they are not promoting them as algae control, but for just oxygenating the water. So FDP is just as glad that I proved it didn't work. And they spent a lot of money trying to find this out. But the company has changed its motto, which is kind of cool. So in water body inactivation, where you add chemicals to the water and tie up the phosphorus, if it's a water body, again, that stays stationary, that's a good thing. It will work. There's phoslock, there's a lot of compounds. Aluminum is your best, aluminum products are the best thing to do it, because when it binds to phosphorus, it gets into the sediment and it stays put. Sometimes it lasts, a few years, sometimes 10 years, 20 years. Alum is another one. You have to make sure the pH is right and other things, and you need an expert to apply it. Adding to a tributary, which is the image on the right, can work. You can add aluminum products to a tributary. But again, this is a continuous operation in the tributary. In a water body, for example, I looked at one from Martha's Vineyard this summer. In April, plenty of algae. They applied it and they sent me samples. They said, we can see to the bottom. And all summer long, it took me forever to do any kind of counts on what's in there because they wiped everything out. And only a few organisms came back, but not cyanobacteria. So we'll see what happens to that same water body next summer. We don't know yet. We got a new project just funded by FDEP where we are actually developing a brand new patented product where we're going to micro encapsulate with like chitosan or other alginate things. We're going to surround aluminum. I'm working with USALCO, probably one of the largest aluminum producers for drinking water. And we're going to encapsulate material that'll float in the water body. So you have to put a little oil in these capsules. And we don't want those capsules to dissolve until it hits the pH of 8.5 and above. That's when blooms are really active. As they photosynthesize, they raise the pH. It triggers, hopefully, the release. This aluminum product that we're going to put in there will precipitate out the organisms without killing them. Perfect. I'm almost done. Anyway, we're hoping that it works. We're still working on the development of that, and we're going to do it in the laboratory first, and we're going to try it in the field. All right, finally, when you go to a water body, make sure you see the warning signs. The lifeguard's not there. The bacteriologist isn't there, nor is the phycologist. Phycologist is the study of algae. For like 10 years, my parents thought I was a psychologist. I said, yeah, I studied the psychology of algae. So anyways, if you're really bored and you want to read it, the Color Atlas of Freshwater Algae, Comprehensive Identification by yours truly, came out last August. It's a coffee table book that they miniaturized. I'm not happy with Springer. I mean, I have it as a big manual for my class. You flip it open. And they made it like this little skinny book. It's barely the size of this thing here. But whatever. My name's on a book. And also thank you. And just so you know, algae are really, this is a natural organism from the Everglades. Look at all the colors in there. If that one isn't enough, that's it. Open for questions, by the way.
thank you. We have plenty of time for questions. If anybody has any questions they want to
I
start.
could go longer.
I have a couple. First question, I remember probably 8 or so years ago, I went to a blue-green algae task force meeting in Naples, and the Surgeon General at the time was arguing that the EPA's recreational water quality criteria was based on ingestion from rats in a controlled environment and wasn't based on some sort of epidemiology exposure system.
Yeah.
And the argument was that the state of Florida needed to develop a better way to calibrate recreational exposure. Do you know where the state is with that? And can you debunk that? I saw your.
Well, what it is, because I was pretty close to EPA at the time, they used children that were in a swimming pool and looked at, they got chlorine showing up in their urine. That gave them a number to start with. Okay. You know, epidemiology, no, we don't, they haven't figured out much about this. Not from ingestion, not even from aerosolization, which was even more potent. But all the work, I got funded by FDOH, and I could not get my cyanobacteria to come out or release their gases or their toxins, not like red tide. So whole organisms were blowing out. I was doing 40 mile an hour winds, and I was blowing whole organisms out of the water with their toxin, but not the toxin. And some of the toxins are hydrophilic. In other words, they grab onto the water, some are hydrophobic.
We're here.
And there's 300 plus toxins in the microcystin group alone. So it's, I don't think we even have that skill set in the state. They haven't pursued it that I know.
So is the state going to adopt the existing EPA recreational?
They have.
They have.
Yeah, well, again, FDOH, if it's spotted, they will post an abundance of caution. So it has nothing to do with the concentration of anything.
Okay, understood. So they're just
Let's
basic.
let's post.
So we're going to continue with the approach. If it's there, we're posting the science anyways. Yes, the science is ready for that.
So I worked on a lake in Washington, 4th of July. The thing is solid blue green. People might recreate it anyways, even though it was posted. I mean, right? But people will recreate. And if you're water skiing, or jet skiing, you have much greater chance of breaking the water, or a kid sitting on a beach versus being in a hose. Right. But it's a lot different than gravitoxin, which they work so easily with the sea salt spray. Not the same thing with the cyanobacteria.
Second question. So we know that Carina brevis has sort of a deactivation temperature threshold or a dormant period, right?
Yeah.
Do we see the same thing in blue-green algae? Because I noticed we still have plenty of blooms in the river today, even though it's cold as heck recently.
You would if you weren't in Florida.
Okay.
So I've seen blooms all year round, Lake Okeechobee, because I've spent a lot of time on Lake Okeechobee. There's always some organisms there, and there's blooms right now.
Right.
Yeah. So the temperature threshold just allows them to grow faster. Okay. That's it. There is no. Some of the ones can fix their own atmospheric nitrogen. I forgot to talk about that. N2 taken right from the air. So I don't care what you regulate on nitrogen on the land. If you want a nitrogen fixing bloom, East River gets those. That has nothing to do with it because they can grab it, the air themselves. 78% of the air is N2. We can't use it. We need nitrogen. You know, every protein in your body has nitrogen in it. There's a whole suite of cyanobacteria, not microcystis, but there's a bunch of others that have specialized cells for fixing atmospheric nitrogen. 60 enzymes, 16 ATP to break down the triple bond between two nitrogen atoms. So, wait, did I say these were simple prokaryotic organisms? Oh, they've been around 3.5 billion years. They know every trick in the book that we can't, we're reading the first page. But they have huge history, genetics that allow them to be successful.
Thanks.
And they're beautiful. Wow, it's almost. I got a sample yesterday from, the Cedar, Sarasota, where they had a bloom, a green bloom. Green, CB. And it turned out it was a green flagellum, which was really interesting. right off a discharge pipe. So things are blooming everywhere. It doesn't have to be blue-green right now. Not toxic.
Okay, we have,
Are you able to get a question from an online caller?
Sure.
yep,
Yeah, it's Joe Kilrain, Cape Coral. We've made some considerable investments in bubble curtains at the mouth of our port here. And basically, we thought that they were doing good. Also, some of the inland lakes and some of the HOAs down here have put in micro bubble aerators. Can you just comment on the effectiveness of them? And then I have one other question after those.
Any kind of aeration where you're moving the water around will disrupt the pattern of what the organisms want to do. They want stagnant waters so they can move up and down. Quiescent waters in the river, any marina where there's quiescent waters, that really supports them. So the nanobubbles or microbubbles people are using, yeah, that oxygenates the water, which is good, and it circulates the water, and that's good. Nanobubble curtains. So if you're coming out of your shower, would a nanobubble curtain stop the water from coming out? I don't know. I don't see it. I haven't studied it.
Yeah.
I'd want samples on both sides over a period of time to figure out if what's getting through, if anything.
Okay, thank you. The next question is, we were contemplating, we have a huge recycle irrigation system here. And basically, we eliminated phosphorus and nitrogen in off-season times. And we thought we were doing a good job on that, but we still have mulching conditions. Everyone mulches and some of the mulch is getting into the system itself. But my perception on that was that the mulch, the nutrient load from mulch is to be able to manage that process as compared to managing the phosphorus and nitrogen directly from fertilization. The bang for the buck is really in the fertilization control. Could you just comment on that?
I don't really know how much nutrients would be in bark, which is, mulch. I don't think there's that much, and it's probably tied up in the biomass. So, but I've never actually looked at any data on nutrient content of mulch, so I can't help you there. But if you've controlled your phosphorus and nitrogen, that's really critical.
Okay. All right. Thank you very much. Appreciate it.
Sure.
I have one more. Dr. Rosen, is there a generic salinity threshold when blue-green algae lyses in the river itself? Because we see bloom sometimes near where I'm at in Fort Myers, and then sometimes there's sort of a line there where it doesn't seem to persist downstream.
Funny you should ask that. And the big bloom that happened in Lake Okeechobee in 2016, the Corps of Engineers funded a project with me to look at salinity threshold, where I was taking samples and looking at how microcystis responded. We went all the way up to 35 parts per thousand. And one of the first things they do is they start excreting more and more mucilage. The colonies are surrounded by mucilage. It protects them. But they get to the point where they're putting all their energy into making mucilage, cuts back on photosynthesis. But our threshold that we found was about 18 parts per thousand. And they start feeling the effects at 10 parts per thousand. If you go to the Leucospermum, a much more delicate organism, but a worse toxin producer, because it makes some of those nasty ones, makes the neurotoxins, much lower threshold. And they also make extra mucilage. It's just what they do. So, yeah, so that one I would say closer to 6, 7, 8. So you kind of have to know your organism. And are they alive at 18? Probably, but they're dying out and unhealthy and diverted from what they normally do. So anything above 10 starts to stress them. But when they stress, you don't know what pathway they're going to go down. Hopefully, not making more toxin.
okay. We have a question from the chat. Is there a marine algae guide that you recommend?
So you're talking about plankton or seaweeds or? So no, there isn't. And believe it or not, it's in my to-do list because I've done the freshwater. I'm doing another version of that. I'm not giving it to Springer. I'm going to keep it. It's about 500 pages. I have students involved in it, and it's gorgeous. And people want that version that they can flip through. A spiral bound, thick paper. This Springer, the pages brand new book came to me, and they were all wrinkled from humidity. Anyway, but I want to do a marine one. And like I said, I got marine samples in yesterday. As a phycologist, I can identify that. It's just not my expertise per se, but that's something you develop. Not good keys for the marine stuff. Should be, but.
Thank you. Another question?
5, 6 years ago, the governor established the Blue Green Algae Task Force. One of the members of the university, Dr.
Yep.
Parsons, sat on that.
Yep.
We came up with a bunch of recommendations, some of which were adopted and everything. I haven't met in 2, 3, 4 years maybe. Have you heard anything? Is any opportunity getting back together? I don't think they've ever disbanded or anything.
Yeah.
Wondering if you ever hear any rumblings. I heard some members suggest we should get back together. I just didn't know if there's any inside track on whether we make some more meetings or anything.
I don't know what they're doing next, but Mike Parsons, who's in my water school with me, said, "Do you want it?" Because when I came on board, the blue-green guy, I said, "No, it's administrivia. I do not want to go to meetings and sit around." And they didn't really. They went after the nutrients. They went after, well, septic to sewer. What can we do as far as the nutrients? Watershed, that's the first thing. And I'm not sure, they don't do the selection of the innovative tech projects that the governor has been paying for. So yeah, the expertise is there. I mean, they've got good people on that committee. They're all pretty much friends of mine, but I don't know what they're doing.
Well, there is a bill this year, probably won't go anywhere, to further implement the recommendations of the task force.
So in 1996, there was a harmful algal bloom task force set up in Florida. I was on it, but I was at the water management district. It was marine and freshwater. And then when they separated and had the marine one, they kicked me off of it. So.
Okay.
Come on, you want to know more? You have to ask the question. You have to if you want to get a good grade. All right. Thank you for having me.